So that brings us to introducing our first company, which is INanoBio. And Bharath, I believe, will be presenting on behalf of INanoBio. Excellent. And to all the companies, we'll have a clicker right up here.
Hello, everyone. I'm Bharath Takulapalli, founder CEO of INanoBio. We are developing a novel nanopore transistor technology to sequence the DNA 1,000 times faster at 1/10 the cost compared to current state of art to democratize DNA sequencing. Here are the 3 grand challenges in healthcare. Preventing future pandemics would need a single test costing $25 that can detect hundreds of pathogens and any number of variants at the point of care. Personalized medicine, right drug for the right patient, is projected to be in itself $80 billion market. Liquid biopsy tests, blood tests for detecting cancer early, are in development. The thing that's common to all this is our ability to sequence the DNA. As a result, DNA sequencing market is growing exponentially and projected to reach $120 billion by 2027. However, current DNA sequencing systems are very expensive and very slow. There's an unmet need for affordable, scalable technology to democratize sequencing systems. Nanopore sequencing is next generation of DNA sequencing technologies, wherein you pass DNA strand through a nanopore and read out the DNA sequence as it passes through the nanopore aperture. INanoBio is developing a novel nanopore transistor technology by combining nanopore technology with semiconductor CMOS technology that's ultra fast and ultra sensitive. We invented a new kind of technology called FENT, Field Effect Nanopore Transistor, supported by DARPA. It's the first of its kind 3D transistor that has a physical nanoscale hole at the center for the DNA to pass through to sense and sequence. Here is an animation schematic of our chip with array of these FENT transistor devices. This is a cross-section image of the device. So it's a transistor where the electron current flows around the whole nanopore at the center. As DNA passes through, the electron current senses the DNA to sequence. This technology is in development right now. We have successfully developed FENT prototypes with 15 nanometer pores on 18-inch wafers working with a semiconductor foundry in US. We have proven excellent transistor performance and speed of over 100 megahertz, which is at least 1,000 times faster than the state-of-art nanopore sequencing systems. We have passed the DNA through the nanopore and have used electron current in the transistor to sense the DNA with high sensitivity and high speed. Next, we will be reducing the pore diameter and sequencing the DNA. We have done in-depth analysis of sequencing market, and INanoBio stands out. We will deliver 1,000 times faster sequencer that is 90% less expensive. We have seen rapid transition from IBM mainframes, large ones, to personal computers, mobile phones. This has been enabled by CMOS technology and scaling per Moore's law. FENT is CMOS technology, and we project that it would replace current large refrigerator-sized devices, which are NGS sequencers, because FENT would be orders of magnitude times faster, smaller, cheaper, and scalable. We have multiple modes, issued patents, new patents in filing, trademarks, and capital-intensive process. That's a barrier. Here is our leadership team, myself, Mukil, who is in the audience, co-founder. Our scientific advisory board includes pioneers such as George Church from Howard University, Chiming Hu, the inventor of FinFET transistor that powers all processors today, JP Millan, who sits on the board of CVS Health, and George Post, Dr. George Post. We have raised $15 million to date to develop the transistor from DARPA and from seed investors. We are doing a $5 million pre-series A to develop to improve the FENT chip that I have showed you and generate pivotal biological data to achieve value inflection. Thank you.
Well, thank you very much for the presentation. I think the technology is very impressive compared to the competitors. Could you talk me through a little bit on the go-to-market strategy?
Yeah. So the current sequencing market, as much as you see news on $100, $200 genome, the real market for throughput sequencers is 10 to 100 times higher. So low to mid throughput sequencing systems, or the users of the systems, are 70% of whole sequencing market. For them, the real cost of whole genomes or genome equivalent sequencing is $2,500 to $10,000 per genome. That's our first product focus. So we are going to release our first product into that and target a 1/10 cost reduction. Sorry, 90% cost reduction, 90% or less. That is, we'll position it at $250 versus $2,500 or higher today. So it's a sequencer that would go to research market initially, but also into CLIA labs in hospitals and clinics for clinical testing.
Okay. Well, the reason why I asked the question is because this is a very, very competitive market with like Illumina is like right in SoCal, and they're very, very big market share there. So I'm just trying to understand your product, understanding that it's faster and that understanding that it's cheaper. But for a research facility or even pharma, there is an upfront cost of replacement of their existing technology for them to be able to make that switch. So I'm just trying to, how do you ease that burden upfront?
Understood. So fair question. Thank you. We have been contacted by almost all sequencing companies and life science companies, over 5 to 10, over the period of last few years. So they are aware of us. We are aware of them. We have not partnered yet. We'll partner a little bit further. That's the idea. To your question, the genome sequencing market is growing exponentially. This is a true thing. So today, how many hospital systems can afford $1 million NovaSeq instrument from Illumina? Not many. Now, if you can make a $5,000 sequencer that can output 1,000 gigabases at $250 cost, you are making every clinic, almost every clinic that has a CLIA lab can then afford it. That's the idea. So we could do COVID-like tests, pan-pathogen tests, metagenomic tests at the point of care in the CLIA facility. That's kind of the new market we see.
I see. And this is just a live conversation. So my pushback there is what have you looked at the downstream pipeline? Like you generate the data faster and cheaper. What's going after like the analysis part of the data? What happened there is the burden is going to be on that hospital system who's now purchasing a machine that is cheaper, but they don't know what to do with the data.
No, very true. So Oxford Nanopore is the first DNA sequencer in the market, as you're aware. Nanopore sequencing fundamentally can do long reads. One of the shortcomings of Illumina has been short reads. Of course, it's been a strength and shortcomings both, but Nanopore sequencing can do both long and short reads. People have proven that. So one of the features would be that, in addition to other features of portability, using at the point of care. I just met another colleague at this presenter at this conference who was using a Nanopore sequencer in field to do some of his work. So we see that flexibility as a huge, feature itself, in addition to the other technical features. And also the data can be uploaded to cloud or quasi real time on the instrument, depending on the application. If it's a let's say a detection application, targeted sequencing application, you could do on chip or on system processing for quick data in 20, 30 minutes or so. But if it is whole genome sequencing or something broader, then you could do cloud-based and basically, again, depending on the application, you have various options. That's the way we would analyze to answer your question.
Thank you.
So from the cloud strategy, are you working multiple clouds? I mean, so not to be locked in for the secondary analysis or tertiary?
We are not there yet. As you can see, we are doing lab-based analysis. The pore size is large right now, where we can parse the DNA and sense it with high speed and high sensitivity. Next is to reduce the pore size and start sequencing the DNA. Any company that can sequence the DNA using a solid state technology, not a protein-based solid state, would scale infinitely. That's the advantage of this technology. So we could go to Intel Foundry and have billion chips made per month for disposable tests. So that's what we are chasing here effectively.
So if I understand your market strategy, you're using the research use only as kind of an initial bridge, but really the upside is on the high throughput clinical diagnostic point of care. From a revenue time horizon perspective, at what point do you expect the major revenue contribution to flip from RUO to those other markets? So I'm just saying from an investor's point of view,
Yeah.
that time horizon can't be too far out or else it starts to lose that financial return. appeal.
Very true. Fair point. What's happening in this space is if you have seen all the other players and Illumina being the major player, no one is anymore doing FDA approved. So it's all CLIA lab based offerings. So once the accuracy is proven and the usability or the assay is developed, any CLIA lab can offer tests based on a sequencing instrument. So to answer your question, it would not be a stepwise. We see it as almost a gradual transition. The initial research market focus is to prove ourselves when we launch, of course, and then gradually start going to the CLIA or point of care testing market. And also, I have not answered one of your questions. The idea is while we do a low to mid throughput sequencer for these markets, specific markets, and also point of care clinic applications, we do have the option, and that's the plan, to out-license Fent chips, supply Fent chips as OEM to the high throughput sequencing market, because that's a different market. Broad Institute, let's say hospital systems, they are huge. So other current sequencing leaders can develop their own sequences around such a technology is the idea. So it's a win-win kind of thing.
Nice presentation. You spoke to the speed and cost advantages. Can you talk more about the sensitivity and accuracy of the technology to date?
So as I was saying, we do not yet have a small enough pore to sequence the DNA. So we are going towards it. That's the next fundraise, right? That said, previously, the whole project started because we had proven that linear nanoscale transistors can detect very small molecules, actually molecules smaller than DNA bases. It's published data. So because nanowire sensors or transistor sensors can detect, that gave us the idea to wrap this nanoscale transistor around a nanopore. Otherwise, this would, So I hope that gives us a confidence that the transistor sensor fundamentally has that sensitivity. Now, the next question is, can you make it? And that's where our effort has been so far, and we have made it and it works great. And then we have also tested the speed as well in a mock test using a laser pulse system. It's very fast.
Well, actually, when I want to just clarify again, when you talk about sensitivity, I'm not talking about necessarily the size, but necessarily the amount or the dilution of your target in general.
Oh.
So in the clinical setting, if we're going to look for variants of unknown significance or something that is pretty dilute in biological matrix, how does it perform there?
Yeah. No, fair question. Sorry, I misunderstood your question. I think the fair answer is it depends on the application. If you're looking at, let's say, liquid biopsy or blood-based, biomarkers, you have to do a priori, depending on your marker, PCR-based target amplification, and amplicons are then sequenced. Now, if you're going to do a saliva-based, let's say, pathogen detection test, and even there, depending on the case, you might be able to detect it directly. But one thing we could say is there is no reason for us to suspect that this will be any less sensitive than any other technology. Because we have already demonstrated at a prototype level, not on the chip, separate prototype level, microfluidics, where we can isolate and enrich and amplify and so forth. So the lab has done that. So there is no fundamental disadvantage here that prevents us from matching or improving or any other. I hope that's fair to say.
Yeah. So you would use existing pre-analytical prep before. Thank you.
Actually, the idea is to even collaborate there, not invent everything in-house. Thank you.
Excellent. Thank you so much, Bharath. Thank you. Excellent. Great job.